Well, welcome everyone. Thank you for joining us today. I hope we have an exciting panel. As the voice of God just announced, I'm Mark Hurlbert, the CEO of the Melanoma Research Alliance, and I think we have an outstanding panel here that we'll get into a deep discussion. You know, I'm really thrilled to be here and moderating this panel. MRA, we've invested $160 million in melanoma research since we were founded just over 15 years ago, and about 90 million of that has been on immune therapies. So really excited to be here, and we'll just do quick brief introductions, and then we'll dive in. So we'll go down the road.
I'm Gina Carruthers. I'm the president of the Prostate Cancer Foundation.
My name is Frederick Finkenstein. I'm the Chief Medical Officer at Iovance Biotherapeutics. Iovance develops TIL cell therapy, which is a kind of cell therapy that starts with the patient's own T cells that are found in the tumors to make a polyclonal and autologous cell therapy product. We just got approval for Amtagvi or lifileucel, which is the first TIL cell product approved, and also importantly the first T cell product approved for the treatment of solid tumors. So two firsts, and we can talk a little more about what's different, why this works, how this works in our discussions. Looking forward to that.
Hi. My name is Anusha Kalbasi. I'm a physician scientist at Stanford, and I run early phase clinical trials in the immunotherapy space, and I run a laboratory that studies next generation T cell based therapies.
Hi, I'm Connie Pfeiffer. I'm the Vice President of Medical Affairs at Immunocore, and we are in the area of immuno-oncology, and we had our first launch of a product, the first TCR therapy, just recently. So.
My name is Sunil Verma. I'm a clinician oncologist. I joined AstraZeneca now about 5 years ago, and I'm the head of oncology at AstraZeneca for medical. It's really looking forward to this discussion. So I started my clinical practice 20 years ago doing lung cancer when there was really nothing. And to see this evolution over the last 20 years of how immunotherapies has really changed many different tumor types, including lung cancer, is quite exciting. So looking forward to how we shape it moving forward.
Yeah, thank you. So just a level set, and we'll just do like a little background on where we at with immune therapy. So the first new immune therapies were approved in 2011, immune checkpoint blockade targeting CTLA-4. This is what Jim Allison won the Nobel Prize for his discoveries in CTLA-4. But the first immune therapies were approved first in melanoma. We've now had several immune approaches approved over the last decade or so. Really exciting to see where we've gone with one immune checkpoint molecule. These are called immune checkpoint blockade, so giving these drugs, anti-CTLA-4, anti-PD-1, now we have an anti-LAG-3, you take the brakes off the immune system. It basically allows your immune system to go in and attack the cancer just like it would a bacterial or viral infection. And it's been like has been said, exciting progress in melanoma, but I think what's critical is it's already being spanned out across many other cancer types. So maybe we'll touch on some of the progress in other fields. Gina.
Thank you, Mark. Yeah, if we look at the CTLA-4 inhibitor that Mark just spoke about, Jim Allison's work was initially funded by the Prostate Cancer Foundation back in 1997. And the hope was that his laboratory at UC Berkeley would develop an immune checkpoint inhibitor that would literally stop prostate cancer. And that was the initial work. The Prostate Cancer Foundation has funded over $1 billion in prostate cancer research since its founding in 1993. And what we found was Jim Allison's work not only exemplifies the value that the Prostate Cancer Foundation has delivered in terms of funding research with young scientists, and then watching the evolution of those scientists and their drug products when they are successful. And then while it didn't work, in 2006 and 2007, when the initial trials were done by Bristol Myers Squibb, it didn't work in prostate cancer. It was extremely toxic. And then we found years later that the Melanoma Research Alliance picked it up and funded research, and then it was extremely successful. We can see that success in many molecules in immune therapy. In prostate cancer, the tumor type itself tends to be cold to immunotherapies. So we've tried numerous studies, both in combinations with agents that work, to see if we can unlock the immune system to make it sensitive. And today we have a number of trials going on that offer hope, but they're all new compounds. So we can look at bispecific T-cell engagers called BiTEs. Research has been going on in that area for prostate cancer over the last 4 to 5 years. And many of those compounds have delivered initial results and then been pulled back because either the efficacy or the safety doesn't seem to match where we're going. Very similar issues to what we saw early on in immunotherapy. What's exciting is at the Prostate Cancer Foundation, many of the drugs that we have in fact funded and scientists we funded have been very successful at taking those compounds into other areas of cancer. So what we look at is we found that we can look at different receptors that we've uncovered in our research or different mutations that occur in prostate cancer uniquely, and those became targets for therapies. So in terms of if we look at our work relative to olaparib from AstraZeneca, which is a PARP inhibitor, not an immune therapy, we see great success. The target for PARP inhibitors came around work from the Prostate Cancer Foundation's funding 2012 to 2014. It led to a major publication in New England Journal of Medicine that said this is a successful target for the treatment of prostate cancer. So it's a continuing process to unravel, to look at what might work. Today, there's research going on, as I said, in bispecifics. There's the STEAP1 trial. Amgen has had very successful results with. We're optimistic. At the European Society of Medical Oncology last year, that data was first shown. We anticipate more of that data coming up either at the American Society of Clinical Oncology in June or at ESMO, the European Society, again in September. So that continues to offer us hope. We're just looking for different mechanisms of action that will actually give us this opportunity to find immune treatments that work against prostate cancer, and it's going to be in combination.
Yeah. I think it really brought up some great points. I think the success so far with immune checkpoint blockades, has, as you said, sort of spun out across many cancer types. Some of the funding, as you said, started in prostate, but immune checkpoint blockade has worked very well in melanoma for advanced cases of melanoma. Before checkpoint immunotherapies, if you had metastatic advanced melanoma, really poor outcomes, probably Sunil, when you were in medical school or in your early days. But now approximately half of patients can be well treated with advanced melanomas with checkpoint immunotherapies, but that still leaves the other half that aren't benefiting. So I think that's what we'll hear on this panel, where some other new approaches have been brought to bear. So I think we'll start, maybe we'll just have you comment, Connie, around the T-cell engagers and sort of what that is. It's a novel approach. Mm-hmm. And then Sunil, I'd like you to weigh in and think about sort of some of the combinations and things that Gina brought up.
Yeah, no, T-cell engagers are basically taking immunotherapy, hopefully, to the next generation. So an ability to target not only the proteins externally on the cell, but the intracellular proteins as well. So that really opens up a lot more targets for us within, the treatment of cancer. So from that standpoint, we have our first agent, which is on the market, Kimmtrak, which is in uveal melanoma, exploring that in other types of melanoma. And trying to, really harness the immune system in a new way. And I think that's what's so exciting now, is that this is really an unprecedented time of innovation in immuno-oncology, in that there's lots of different ways, there's lots of discovery happening, and I think that's only going to accelerate in the future. So, from our standpoint, it's a new way of harnessing the immune system to really target melanoma, and hopefully in the future other types of solid tumors.
Yeah, I think that's great. Kind of what I think is exciting. Checkpoint immunotherapy, you take the brakes off the immune system, and this allows the immune system to rush in and kill the melanoma or the cancer cells. It also comes with some side effects, that we've been able to, clinically, clinicians can control a lot of these, but it causes some autoimmunity, thyroid problems, a few other things that have been worked out of how to control them. I think what's exciting about this bispecific T-cell engagers that you have on the market for uveal melanoma is it has a specific protein that they can target to the cancer cell, and then it brings in the signal to bring the T-cells in a little bit more targeted, we hope. And
right. It's a more targeted approach, although you can still target some GP100, expressing melanocytes, but you don't get the full body immuno reactions, So We don't see the same type of adverse events that we saw with checkpoint inhibitors, which can be permanent and can be long-standing. With our agent, it's mainly in the first several doses that you see the adverse events as it's really activating the immune system. And so that is in a way a safer alternative and a more targeted approach to immunotherapy. So it's really refining the way that we utilize the immune system against the tumor cells.
Yeah, absolutely. And I have I've personally counseled and met with many patients with uveal melanoma that have benefited from the agent. And it's exciting to see where you'll go with other cancer types. So, but Sunil, maybe we'll have you weigh in, and then we'll get into some cell-based approaches.
Yeah, no, I'm happy to talk a little bit about sort of combination strategies. So certainly, as was mentioned, the wave of immune therapies has really changed how we think about sort of tumor targeted therapies, and it's really improved outcomes. But one important element is one of the reasons why immune therapies tend to be very effective is in response to expression of what we call neoantigens. So is there a way that we can increase that expression of neoantigens so that the immune system is more empowered to be able to react to that? So one of the traditional ways of looking at that was using existing platforms like chemotherapy, where you combine chemotherapy with immune therapy in order to be able to harness the power of both of these modalities quite significantly. But there's limitations with chemotherapy. One of the big limitations with chemotherapy, of course, is not only does it target tumor cells, but it can also target lymphocytes, including T cells, and that may actually decrease the effectiveness of these T cells to be able to engage in this process. So one of the things that we are quite focused on is antibody-drug conjugates in AstraZeneca. And the concept with antibody-drug conjugates is that you deliver chemotherapy right to the tumor cells and the cancer cells. And thereby you have sort of this lymphocyte sparing effect, and you have a targeted kill of the tumor cells. And if you can do that, you may be able to synergize that with immune therapies more effectively. So this idea of combining a targeted chemotherapy with antibody-drug conjugates with immune therapies then could be harnessed very well together. We saw some data, and I think in GU malignancies at ESMO, where it was just mind-blowing when you take a look at survival in bladder cancer when you combine an antibody-drug conjugate with immune therapy. And we have shared some data at AstraZeneca, early data on phase 2 setting in breast cancer and in lung cancer of combining these two modalities and showing that you are maybe able to further enhance the effectiveness of immune therapies when you give a backbone of antibody-drug conjugates. So we are really excited about this combinational approach, and if it can really further progress the benefits that we have seen with immune therapies in a broader patient population, and potentially even in diseases such as prostate cancer and breast cancer, where traditionally immune therapies have not had as profound an effect. So I think this is another way that we can expand the patient population that can benefit from this class of drugs.
Wonderful. I think we should touch a little bit on combinations, and I don't want to leave out molecularly targeted therapies. So for those that know melanoma well, about 85% of skin cancer, skin melanomas, are driven by mutations in a pathway called the MAP kinase pathway. About 50% of these are BRAF mutations, and then NRAS and NF1 loss. And I don't know, Sunil, or if one of the other panelists wants to comment, but in these targeted therapies, BRAF MEK inhibitors were first brought to market around 2011, 2012. But fast forward, in just a short amount of time, we've actually had some large clinical trials talk about when best to sequence them, and when do you give an immune therapy versus a BRAF or MEK combination targeted therapy. So I don't know, Friedrich or Sunil, if you want to comment on sort of the combinations and sequencing.
I think sequencing is a really interesting topic here, right? It's biologically and scientifically interesting to see what kind of pathways do you want to block, or what kind of mechanisms do you want to pursue first for the benefit of the patient, but also for the long-term benefit of the patient. And that this is exactly what was really interesting in the checkpoint inhibitor, BRAF inhibitor story, where we found that patients over the long term benefit better if you do checkpoint inhibitors first and then BRAF, rather than the other way around. It's counterintuitive that it shouldn't be the same, but it is not. And I think it's really interesting to think about why that is, and folks are figuring this out. But I think it's also interesting in the context of seeing how things are moving across the treatment landscape, where, again, you brought up the chemotherapy combinations, right? I was part of bringing nivolumab, a PD-1 inhibitor, into lung cancer treatment, and at that time, we brought it into the setting after patients had progressed after standard chemotherapy. And we thought, because this was based on T cells doing their job, you can never ever combine this with chemotherapy because the T cells are damaging, the chemo damages the T cells. It's not true. Actually, the combinations with chemotherapy are now being used broadly wherever chemotherapy is part of the first-line therapy for advanced cancer in combination with checkpoint inhibitors, and it works. Important now, though, is that leaves an empty slot where the checkpoint inhibitors were before, which is after frontline therapy. What do these patients have available to them now that they progressed after both chemotherapy and checkpoint inhibitors, which have been given in combination? And that's where we're looking for additional and alternative and differentiated mechanism of action, and that can be other targeted therapies, that can be novel chemo, and that can be cell therapy, which has been somewhat of a hard nut to crack for solid tumor diseases, to bring cell therapy into that setting. And whenever we're ready to talk about that, we can talk about the activities and some of the challenges and what to do about it.
Along the lines of when to do these immunotherapies, I think one of the elephants in the room is disease burden, and so chemotherapy helps us reduce disease burden. The immune system, I don't think, was evolutionarily developed to handle billions and billions of cancer cells or even viruses, and so being able to use immunotherapy in a setting where you have a limited disease burden, I think, is important. I think that's possibly what is bearing out in the trials with combination chemotherapy. But we're also seeing that even patients earlier than that, before they even get to chemotherapy, may benefit from getting immunotherapy up front in locally advanced disease before the cancer has spread. For example, in melanoma, giving immunotherapy or immune checkpoint blockade before surgery may actually have advantages, even advantages compared to giving immunotherapy right after surgery. And so as we learn about this, I think people will be using immunotherapy earlier and earlier, though we still have to be cautious because immunotherapies are not without side effects. And in patients who can otherwise be cured, giving immunotherapy up front and giving them lifelong toxicities when they would have otherwise been cured with surgery may be more concerning than somebody with metastatic disease. So I think these are kind of all in line, the timing, when to use it, whether with chemotherapy or earlier in disease, later in disease. These refinements, which may be incremental improvements, but I think they will affect lots of lives because many patients are on these therapies now.
Can I maybe just add something to the targeted element, which is, I think, really interesting? So, in biological principles, when we think about targeted therapies in cancer, as you were mentioning, Mark, you think about sort of those drivers and those mutations that drive, and you target that with the right therapy, and the concept of how targeted therapies have evolved in cancer therapy. And I think it's important to denote that in immunotherapy, we do have tumor agnostic, so that it works across any tumor, a signal, which is microsatellite instability, MSI high. So if you have that, irrespective of the tumor, that these cancer cells are going to be sensitive to immune therapy, which is, I think, a really unique way of thinking about how we can look at, irrespective of the biology of the cancer, that you have a drug that's going to be very effective. So I'm really interested, as we start to expand this thinking about how we evaluate the next generation of immune therapies, of coming up with the right biomarker that is more applicable irrespective of the tumor origin. So I think that is also an important frontier as we think about the next wave of therapies, of how we think about immune therapies in a more targeted manner for the right biomarker selected patient population.
That brings up another really, really interesting topic, which is the heterogeneity and the patient-to-patient differences, right? These microsatellite instability is basically a measure of the tumor's ability to generate lots and lots of mutations, and some of these mutations generate proteins, either intracellular or expressed at the cell surface, that are being shown to the immune system and recognized. And we call those neoantigens. And the interesting piece here is that the vast majority of these are patient-specific, because they're random mutations. So you will find very little overlap in what a individual patient shows their own immune system to the next patient, maybe even with the same tumor type, melanoma or prostate cancer, because of this. And that is what we really need to think hard about, is how do we go about it? How do we use targeted or non-targeted therapies? How do we use therapies that go after specific antigens versus therapies that go after a set of defined mixture of antigens, maybe even patient specifics, or something that is somewhat antigen agnostic, like the TIL cell therapy platform that we are using?
I think one of the things that's fascinating about what you just mentioned is if we look at prostate cancer, which I described earlier as basically an immune cold tumor, when we look at MSI high, as these patients are thought about, we have two serious challenges because immunotherapies haven't traditionally worked in prostate cancer patients. We don't have as many medical oncologists who are looking for these mutations. And one of the characteristics in prostate cancer that's very important to note is these patients decline extremely rapidly. We go through our traditional therapies of hormone therapy in combination with other androgen-targeted therapies. We go through chemotherapy. It's a very fast-progressing cancer, and we need medical oncologists to stop and recognize that then may be an indicator that this patient would respond, in fact, to what is approved, which is pembrolizumab. And there's a few patients that I've worked with who, in fact, are cured, or at least cured for more than 5 or 6 years with no further progression of tumor. And so the key is to identify those individuals, because that's now what you're talking about, as opposed to the tumor itself, right? It's that combination of rapid mutations that then aren't responding to traditional therapies. And this is life-changing in terms of how we treat them successfully. And it's really a huge education challenge, because we need to train medical oncologists, not just in academic centers, but in private practices. And because these therapies often have, the immune checkpoint inhibitors have side effects, they're not necessarily in the urology space trained. And many of these men are first line being treated by the surgeon for their medical treatment. So that's kind of a real world phenomenon that we have in prostate cancer that's different, I think, than other tumor types, where the patient is often being seen by a medical oncologist. So 80% of our men are being treated in the community, and they're oftentimes being treated locally by the urologist, the surgeon, because they're still in the hands of the surgeon, because these diseases are progressing at a cellular level, but they're not identifying why they're going so quickly with active treatment.
I think that a lot of good points raised. I want to come back in a minute and, Anusha, just have you describe what you meant by disease burden so the audience all knows what we're talking about. But just to sort of, highlight sort of what we've talked about so far. So we've talked about checkpoint immunotherapy. We've talked about these new bispecific T-cell engagers that bring the immune cells right in. And I think what's exciting in the field of all of oncology, not just that we have these checkpoint immunotherapies in melanoma, in bladder, and other cancers, but I think, Sunil, your point of this sort of pan-tumor marker, microsatellite instability, it's a marker that could be any cancer that has a lot of instability, is a marker. And I think, again, sort of these immune checkpoints was the first ever sort of pan-tumor approval, right? With PD-1 inhibitors that are now approved for not just melanoma, but any tumor that has MSI high. I think that's an exciting thing. But Anusha, could you just clarify what do you mean by disease burden and sort of, but more importantly, what are you thinking as far as like when best to intervene with some of these immune therapies? So.
Yeah, I mean, patients come with different amounts of cancer in their body. Some patients come with one centimeter tumor. Some come with dozens of 2 or 3 or 4 centimeter tumors, or bulky 10, 15 centimeter tumors, and they present different challenges. And so when we design therapies like immune-based therapies, and if we're relying on the patient's own immune system to be able to expand and attack these cancer cells, you're asking a lot. You're asking a lot of a patient's immune system to basically T cells fight cancer cells in a mono fashion. So having billions of T cells being able to eradicate billions of cancer cells is a lot to ask. And so there are other approaches potentially to engineer T cells outside the body so you can give these large numbers of T cells to patients to be able to eradicate the tumor. Or approaches to kind of reduce the amount of disease, like combining with chemotherapy, or using these immunotherapies earlier in the stage of disease. And I mentioned that some of the patients with melanoma seem to benefit when that's given earlier rather than later. So depending on the state of the disease, the type of immunotherapy is going to be different in what we choose.
Can I add to that, Anusha? Like, I think it's a really important principle of trying to understand when is the right time to interrupt or intersect the disease. The drug development paradigm, however, is traditionally thought that we should evaluate these therapies in a very late line setting, right, where we have patients who have exhausted all standard of care, and then these patients come in their 3rd, 4th line metastatic advanced stage setting, and then we say, all right, let's evaluate this new drug in this patient who has been resistant to everything else to see if it works. And then we will declare success whether patients respond to this treatment or not, and if they don't respond, we're going to kill this drug, which is completely opposite of what we should probably be doing in thinking about how we evaluate some of these therapies when you have a unique armored tool like the immune system, but we're putting this tool in a battlefield that is just completely unfair. So we need to think about when is the right setting for us to evaluate this tool. And I think to your point, it could be in the early stage setting when the tumor burden is low. And I think we need to start to work very closely with investigator, with regulatory agencies to be able to deploy some of these studies in a different evaluation paradigm than we have in the past to really exploit the power of these tools in a more fair manner and on a more representative manner of where they're likely going to be successful.
There's another reason for why we do that, though, right? Yeah. So I think one needs to be very clear. It is obviously wanting to go into a population where there's unmet medical need. Sure. These patients are oftentimes in a position of where they are willing to join clinical trials. Number one, because that is a way to get access to novel therapies. Number two, and many patients do consider that because they want to help out, right? They want to volunteer and help generate data. For us drug developers, we start in that setting often because we want what we call a clean signal, right? Because we administer a novel agent in that setting where we do not have to combine it with a standard of care available therapy. Because once we start doing that, things become kind of complicated because we need to then tease apart the effects of the combination partner versus the effects of the novel agent, and ultimately the regulators who we are submitting these data for review and potential approval for too, they will want that controlled. And that means running what we call controlled randomized trials, which is a big effort and a big undertaking, particularly for smaller companies that have something new they want to bring to fruit, right? So I think that's the other reason for this drug development paradigm, in addition to obviously questions of unmet medical need, having to establish safety first in a population that does not have alternatives and is willing to take the risk. It's complicated, but it's all doable.
I think the opportunity, however, if you were to take a look at, and Anusha mentioned, our traditional paradigm has been that the drug delivery or the drug evaluation is driven by medical oncologists, where medical oncologists tend to be very engaged in that late line setting in order to be able to assess effective therapies. But as the paradigm, the care paradigm is changing in cancer with multidisciplinary teams with radiation oncologists, surgeons, medical oncologists right at the beginning, I think there is an opportunity for us to evaluate how we can do drug development, specifically with immune therapies, in these window of opportunity trials where you have an early signal. You're not delaying access to standard of care, but you're having surrogate markers to say, is there a benefit in a more homogeneous patient population? And I think that requires a reshift to make sure other disciplines are part of the clinical trial paradigm than the traditional, very medical oncologist driven. So I think there is an opportunity for us to do that, because I think the next wave of therapies will need to be evaluated in parallel in a late line unmet need situation, but also in a more homogeneous situation that's probably reflective of early signals.
To that point, to that point, I think it's interesting in the field where urology and medical oncologists have come together, and this trial was very specific to renal cell carcinoma. And the goal, it was a neoadjuvant trial called PROSPER, where the delivery of nivolumab was preoperative, and then working with the medical oncologist postoperatively to see how did that work. And the patients received 8 cycles post-op of nivolumab. So it was a single, not combination. And I think there's questions because at the end, the trial was deemed to fail simply because these were stage 3 renal cell carcinoma patients, and it didn't change the progression of the disease. However, when you look at that data on individual bases, you start to see that there were people in that trial whose tumors were extremely aggressive, where the tumors themselves had wrapped themselves around the vasculature that is part above the kidney. And in those patients, those tumors shrunk back, allowing the surgery to be done without having to touch the vasculature. So in these surgical cases, one of the challenges with the surgeon is it's not just the urologist who's dealing with removing the cancerous tumor, but then you need a vascular surgeon to rebuild the vasculature. And what we don't really understand is what kind of seeding of the cancer is taking place. So in these very complex cases, surgical cases, men had very positive outcomes at a very small number, right? Because not all of the patients had the same tumor burden, and that's one of the big challenges. And of course, that wasn't one of the entry criteria. But when we look at that, I think we think of it as very exciting, at least in the genitourinary space, of what can that mean for men and women with kidney cancer.
Connie?
I think what kind of comes out of this discussion is that we have to challenge ourselves to be innovative in the way that we evaluate new molecules, right, and change the way we do clinical trials. We also have to think about, like, are endpoints, adequate, especially for immunotherapies, right? We're used to looking at, the response rate, progression-free survival. Are there better, surrogates of someone who's going to be, benefiting from these therapies? We saw that with checkpoint inhibitors. We certainly see that with the bispecific TCRs, that, disease control, these may ctDNA response, these things may be better at predicting those patients who are going to benefit or are benefiting and are going to have longer overall survival. So I think we're at the precipice of a lot of understanding of where things are going, changing and innovating the way that we're evaluating our therapies, and also understanding more of what's happening at a tumor microenvironment level, which is really going to help us with combinations, is going to help us with sequencing, knowing those patients, that are going to do better. So, we have some data that we're looking at a specific T-cell signature, maybe that predicts. So I think there's a lot that we don't know that we are on the precipice of beginning to understand, and that's hopefully going to help us in answering a lot of the questions that people here have brought up.
I think you're spot on, Connie. I think you're secretly advertising. There's another cancer panel on biomarkers and blood-based testing, I think later on tomorrow. So, but I wanted to shift gears, but before I did, Sunil, you brought up a great point about the multidisciplinary team. And in melanoma, I remind us all dermatologists, we even have intralesional therapies where they can inject a therapy right into the mole or the melanoma cell. And so engaging dermatologists, surgeons, and the whole spectrum, not just medical oncology, I think is key. I want to shift and talk about cell-based therapies and then also vaccines. And then remind the audience, if you have questions, you can enter one on the app. We've already had a few come in. Or if we have time, we'll let you raise your hands. But before we dive right into cell-based therapies, so we have these drugs that can release the brakes off the immune system. We have these other new drugs that can bring the T cells right into the tumor. But what about the cell itself? And how about we just deliver the T cell right to the patient? So why don't we start with you, Frederick, and then Anusha.
Sure. Let me just plug that we've also had drugs that were the gas pedal, right? I think the first immunotherapy drug ever being clinically approved was interleukin-2, which was really a gas pedal, right? But at the same time, the observations with that particular drug kind of created a first paradigm of an immunotherapy-sensitive tumor type, right? Which for decades was renal cell cancer and melanoma. Yeah. And the checkpoint inhibitors really were the paradigm shift where all of a sudden we now saw benefit in many, many tumor types, almost no tumor types where you could not find a patient population that would not benefit from checkpoint the therapy. Then I think I'm coming to the cell therapy.
Yes.
Then I think the next paradigm then was how to build a cell therapy, and the first generation of cell therapies were the engineered cell therapies, right, where you basically taught a T cell to recognize something different from what it was originally made to recognize. By transducing a new receptor into it, and that could either be an original T cell receptor, and that made a TCR T cell, or a chimeric antigen receptor, which was the CAR. Both of those are being basically plugged into the T cell in order to identify one specific antigen, and that worked really, really well for certain hematologic tumor types, B-cell malignancies, because you could target a target that was basically a lineage marker. It's a target, CD19, that's expressed on all B cells, the good ones as well as the bad ones, which is not a problem. You can eradicate all of them and then just support the patient with something external that helps fill the gap of the patient not having any B cells. That now created a new paradigm, which is, cell therapy works only in hematologic diseases because that's the only place where you can do this. Solid tumors do not have these lineage markers that you can just generally use to eradicate that, and they are more heterogeneous. There's different antigens presented by solid tumors. Even within the same patient, a tumor expresses a lot of antigens, and sometimes even from primary tumor to metastases, not necessarily the same antigens are expressed. So the paradigm was cell therapy does not work in solid tumors because of that, which was a real bummer because solid tumors are 90% of patients' malignancies. Only 10% are heme malignancies, lymphomas or leukemias. And now folks are developing cell therapies either by looking at very specific tumor types or subpopulations of tumor types, and are looking for these tumor-specific antigens that they can go after. That's shared with ADCs are doing that. T-cell bispecifics are doing that, and then people remembered that there was something that actually had been around for quite a while already, and developed by Dr. Rosenberg at the National Cancer Institute, which was called TIL cell therapy. TILs are tumor-infiltrating lymphocytes, meaning T cells that sit in the tumor, And the majority of those are there for a reason, because they want to fight the tumor. But obviously, since the tumor was there, we're not able to do what you just said earlier, which is really at the early stage, avoid that a tumor actually gets formed and cancer arises. TIL cell therapy takes a piece of tumor out of the patient's body and then grows the T cells that are found in that tumor biopsy into a T cell product that is specific for this patient, for this tumor. The manufacturing process makes billions of cells. They are, we call them rejuvenated, because we're taking off the brakes of these cells by taking them out of the tumor microenvironment, which oftentimes is immunosuppressive. And they're polyclonal. They have lots and lots of specificities. And we believe that is one of the reasons for why this approach works in solid tumors, while using CARs or TCRs remains challenging. So, sorry for this slightly longer story, but it's a really interesting story to see how you move about and switch your approaches, because the challenges are very specific when you try to do something new and innovative. In this case, it was a technology that was around and was kind of stuck in academia, and it took a lot of work to take it out of that space and make it work, so you can make it into a medicine. I just stop here.
Yeah, I think this TIL approach, I mean, what's so exciting for patients with melanoma and other cancers is taking it and making it a drug or a product, taking it out of academia. And, and specifically, many patients with some rare melanoma subtypes, mucosal melanoma and others now, are benefiting from the TIL therapy. So really exciting to see where it's been applied and where it will go moving forward. Now, Anusha, I was going to ask if you could sort of describe CAR T in a little more detail than Friedrich has, and sort of how it's different than the tumor TIL.
Yeah, just to give a little context, the way I think about these treatments is either you're a patient that has an immune system that can see and fight your cancer, or you're a patient that doesn't. And that's maybe oversimplified, but immune checkpoint inhibitors and TIL therapy require that the patient already has T cells that see and recognize its cancer as foreign. And a lot of patients just don't have those T cells. And so we have to be creative and think of ways to make T cells that now recognize and see the cancer. And thankfully, we have incredible genetic modification tools at our disposal now. They've been around for decades, but they're getting easier and easier. So we can take T cells from a patient's blood and take them back to the lab and modify the DNA so that the cell does all kinds of things. We can make the cell see a cancer. We can make the cell more fit. We can make the cell expand. We can make the cell get into tumors. We can do all kinds of modifications. And there's a whole next generation of modifications that are coming. But the original modifications were just, let's make the T cell see the cancer. And those are CAR T cells, essentially, but also TCR T cells. But CAR T cells, like Friedrich mentioned, are a resounding success in leukemias and lymphomas. But in solid tumors, they were not working for many reasons. But I don't think that's what's happening at this moment. In the last just 1 or 2 years, we've seen trials where in solid tumors, CAR T cells can be effective. The challenge is that you have to find a tumor that expresses something on its surface that is expressed in almost all the cells, or most of the cells. And there are tumors that have those targets. GD2 is a target in brain tumors of children. And there were some really exciting results using CAR T cells that take children who are otherwise in the ICU and basically eliminated their neurologic symptoms in a short period of time from an infusion of CAR T cells. In gastrointestinal cancers, there's a claudin target that has shown promising efficacy. So there are signs that CAR T cells can work in solid tumors. And I think it's important to remember how long it took us to get to this point and not to be discouraged. TIL therapy, I worked in Steve Rosenberg's lab in the mid-2000s. And at that time, TIL therapy was kind of a niche thing on the side. But they had been working on that for about 20 years and hadn't gotten anywhere in terms of widespread adoption or even industry adoption. But now we have not only TIL therapy, we have CAR T cell therapies, and all of this has happened in about 10 or 15 year period of time. I think now with the new tools that we have to engineer T cells in the lab, I mean, the things we can do to modify a T cell, tumors that, they're mice, but what we could never imagine curing in mice is now just easy. I mean, all the labs, academic labs in the country are publishing papers on curing mice with T cells that before this was not possible. And now those things are now emerging into the clinic. So we're engineering these T cells that are incredibly powerful fighters. Now the question is, are they going to be too powerful? Are they going to cause toxicities? Are there going to be issues? Are we going to be able to control them? Are we going to be able to regulate them like remote controls in a patient? So kind of futuristic it sounds, but I think CAR T cells allows us a platform to be able to do these kind of modifications in patients. So actually with funding from the Melanoma Research Alliance, we're running a phase 1 study of CAR T cells in patients with melanoma who have relapsed after immune checkpoint blockade, with the idea that the reasons patients respond to immune checkpoint blockade may be different from the reasons they respond to CAR T cell therapy. So the patients that fail immune checkpoint blockade may actually benefit from CAR T cell therapies. The challenge with that is patients get further and further along in their disease course, so their disease burden is more difficult, their tumors are more resistant. So it's a challenging space, but it's an opportunity. And I think with the modifications we have in the clinic, it's possible. The other thing I want to bring up is the other challenge with modifying T cells is that it takes about a month or even 40 days. And so a patient has their T cells removed from the body, and there's this 40-day interval before we can actually have the T cells ready to go for the patient. And so there's a lot of progress being made in how to shorten this interval, make genetic modification faster, and there are companies developing tools, CRISPR-based approaches, and those are exciting. But then there's another avenue of maybe even actually making the T cells inside the patient itself. So by directly modifying the DNA of patients' T cells within their body and eliminating that 40-day window altogether. And so I think that's something that I personally think is a very exciting approach to make this a more feasible and accessible treatment for patients.
Interestingly, we are back at the same story that we were with our systemic drugs in modifying these T cells, right? You mentioned obviously the engineering a target specificity into the cell is a big engineering feat. But you also mentioned other things that we can do to make these cells more potent or safer, right? So we're back at playing with gas pedals and the brake pedal, right? Exactly. We're removing genes that code for inhibitors of T cell function, and you can do that in a CAR T. You can also do that in a TIL. Or we're engineering in the gas pedal, where we basically have these cells bring their own cytokine, their own growth factor with them. There's a lot of stuff going on, a lot of modules to mix and match and optimize things. It's really exciting time.
So I want to turn, we have a long list of questions that have come in from the audience in the room and online. And this gets to some of the things we talked about in some of our prep calls, is sort of the delivery of these amazing technologies to patients and access. And one of the first questions was, can someone explain about the state of the supply chain, manufacturing and transportation of immunotherapies? So can one of the panelists weigh in on this?
I'm happy to get started, and please jump in as well. So I think, as was highlighted by Anusha and Frederic, is you can imagine this process from identifying the patient that we need to be able to get their T cells, to be able to do this whole process of engineering, manufacturing, expansion, and then reintroduce these T cells back into the patient, what we call sort of this vein-to-vein time, can be anywhere from 6 weeks to 8 weeks. And then you need the manufacturing capacity to be able to actually do this. So this is a significant challenge across multiple different pharmaceutical companies to be able to maintain this degree of supply to be able to reach the demand that's emerging. The second challenge is, which are the patients who will actually drive this benefit in which institutions? A lot of those patients are being served by academic institutions. So, and as Gina, I think you mentioned, most of our care that we see, at least in the US, is in the community setting, where 80%, 70% of patients are being treated in the community setting. So now we have disparities that we're introducing because they're going to academic centers, they're being identified, and the care that's happening in the community is not the same. So a big opportunity that we have is to really sort of speed up the process, make it more off-the-shelf process, or make it sort of in vivo process to be able to deliver this and make sure that you have access in the community settings to be able to deliver this effort. And I think that is certainly something that we are working quite hard on, to be able to expand those centers who can deliver this, to be able to reduce the time from identification of patients to delivery of this medicine so that it's feasible. Because patients cannot wait for 2 months when they have rapidly progressing cancers, specifically in the solid tumor setting, and patients cannot be treated in a very distinct manner when they are academic or in the community. So those are the challenges that we are addressing in parallel to anticipate how these drugs are going to be reaching and serving patients in a broader and a more equitable sense.
I think important points. A second question is around how are we getting over T-cell exhaustion and in the manufacturing process? I don't know if you want to weigh in, Friedrich or Anusha, about T-cell exhaustion.
There's several ways to do this. One is to shorten the time that it takes to make the cells. T cells, what is T-cell exhaustion? It's a way of saying T cells get tired of trying to kill and tired of trying to grow. And T cells tend to work better when they're earlier in their lifespan. So this earlier we can capture a T cell in something we call stemness, which is a reference to them being more stem-like or stem cell-like. The earlier we can use T cells, the more functional they are in eradicating tumor cells. And so there's lots of ways to do this now. We can use drugs that prevent the T cells from going down that path of exhaustion. We can use proteins that signal the T cell to avoid exhaustion. We can genetically modify the T cells to prevent exhaustion. So there's a lot of tools. One of the challenges is which ones do we bring to the clinic? There are so many approaches now in various academic and startups around the country that one of the questions is, how do you test all of these? And there's questions about, do we do competitive tests within a single patient where we give the same patient 5 different modifications and see which T cell wins? And would the FDA allow us to do some kind of competition within a single patient to get to the answer faster? Otherwise, there's no way we can possibly screen all these approaches that are impressive within preclinical studies.
There's not just that. There's also, how do you control it? How do you reproducibly have it in a controlled way so that the regulators are comfortable with what you bring forward as something that what we could consider is a controlled drug? Meaning, how do you demonstrate that the drugs that you are producing when you are making a commercial product is the same as the product that you use during the clinical trials? That is what the CMC, so chemistry, manufacturing, control, does, and what these famous release assays are there for, right? That is what takes a lot of effort, a lot of very smart people, and a lot of experience. And I think that is what takes taking a technology like CARs or like TILs out of the academic space into the biotech or industry space, because that's where the firepower is, that's where the resources are and the expertise are to make that happen. And it's going to happen more and more. And the regulators are going to learn in parallel with the folks who are doing this.
That's important. I was going to ask Connie if you have any comments about sort of manufacturing and getting the product to patients from Immunocore's perspective.
Well, we're an off-the-shelf bispecific, so we don't have some of the challenges in the fact that you can order a vial and treat the patient immediately. The thing that slows our process is not so much the manufacturing, but the testing the patient to make sure they have the appropriate HLA type. But that, generally does not take more than, a week to 2 weeks. And then, we have a cold chain approach, so it's just keeping the product in a certain temperature controlled approach. So we don't have the same issues, but obviously we're going to see some innovation in that space, hopefully an off-the-shelf eventually there.
I think you've all touched on really important topics around sort of the off-the-shelf versus customized to the patient. How do we manufacture and deliver these? not just to the top academic centers, but all across the country and hopefully all across the world eventually. A few other questions have come in. I mean, actually so many, so apologies if we can't get to all of them. But a few people have asked about other cancers. So could anyone comment on pancreatic or head and neck and any progress in immune therapies in these cancers?
I can maybe touch upon the solid tumors that we have been sort of focused in on. So there's targets which were identified, I think Anusha mentioned, in brain cancers. But in addition, STEAP2 in prostate cancer is an important target that's a number of companies, including us, are evaluating CAR-T. We also have Claudin 18.2, which was mentioned, which is in gastric cancer, but also some expression that is seen in pancreatic cancer. So there's an opportunity there. And then there's GPC5, which is in hepatocellular carcinoma, also a really important target. And we'll be sharing some data at the upcoming ASCO meeting with our CAR-T for that. So I think there are many of these tumor types. And I think, Frederick, you mentioned what's unusual about solid tumor is that you don't have sort of this lineage-specific phenomena. You have polyclonality, so you have multiple different targets. So I think one opportunity is you can maybe reduce the disease burden by giving an induction therapy, such as with chemotherapy and antibody-drug conjugate, where you decrease the volume and then you see which is the right target to go after. So I think this is maybe combination, but more in sequence to be able to leverage that phenomena so that you have a lower disease burden and a higher likelihood of success. And that's certainly the approach that we are considering as we evaluate CAR-Ts in solid tumors.
From our standpoint, our MTAX platform, we can change the target or the effector end. So the effector end is more that you could see it be used in other areas other than oncology. But the target, we have a PRAME target molecule that we are looking at other solid tumors. Head and neck is included in the phase 1 study. So from that standpoint, we're hoping we're going to see applicability in a broader range of tumors from PRAME. We have a PWIL product that's, or molecule, I should say, that's coming, through our pipeline. So it really, we hope, is going to give us a broad platform whereby we can develop molecules that will then target a variety of different, tumor types and have benefits. So.
Yes, sir.
I just want to tie in a couple of these comments. Same question coming from different sides. And Mark, I thought you were going to ask my question. But I haven't heard any of you talk about, so you have patient outcomes. So to your point, you got a non-aggressive tumor, you move surgically, readily treated by chemotherapy. Right. You're never going to get to one of these more advanced ones because your patient requirements for best patient outcome is simple, right? On the other side, you got some of these things that Frederick, you were talking about. I'm thinking primarily some of these super expensive T-cell modification therapy. I think the first 6, like 100% efficacy, but $1.4 million per patient, impractical from a medical practice perspective. So how do you combine these two issues where you're not going to really see the advanced ones because they're only going to go to your next wave, and I know this panel's on next wave, after surgery doesn't work, after chemo doesn't work, and then secondly, you've got a really high degree of efficacies that maybe if you continue to process those, your 1.4 million would go down to $40,000, and now it is an economical treatment.
So I'm happy to take it. So I'll put sort of a breast cancer clinical experience because we have faced this and addressed this in breast cancer, I think, really well. And there's always this evolution in cancer care treatment, which we're seeing in hematological malignancies in breast cancer, where you bring a range of therapies that a patient can benefit from, and then you start to ask a question, how do you delayer, how do you de-escalate the treatment? So in breast cancer, for example, we know that the 5-year overall survival for early stage breast cancer is 85%. They do really well. And we also know that there is a certain portion of patients, up to 50–60%, who will do very well with just surgery alone and who may need a pill, a hormonal therapy that's very well tolerated and that's cost effective. So I think that layer of when do we de-escalate and when we escalate is a question that is prevalent across all tumor types. This is not going to be an answer for every single cancer patient. This is really for those resistant patients where the current therapies are not going to be effective, or identifying it much early on where we predict that the current therapies, the cancer will be resistant to the current therapy, so we give the patient the best shot to begin with. What happens in many cancers is that when the cancer goes from early stage to metastatic, it starts to become incurable. So part of this assessment is to say which are those patients who are most likely going to be benefit, where you have the best shot at cure in the early stage when the tumor burden is low and you have the right approach. Not all patients, but really those patients who really need that therapy upfront. So I think that is part of the assessment in order to make sure that we personalize those treatments.
I think another thing is that advocacy organizations, such as we have, representatives from here, getting that patient voice back in about the effect on them, the financial toxicity to the system, et cetera, is really, really critical, and making sure that we in the pharma industry engage, with those groups, because it gives us a different focus, a different point of view. So I think making sure that we have strong relationships there is really critically important, and making sure that the patient voice is throughout the development process.
I think that's a good note to end on. The patient is all of our North Star for all of us.
The end of everything we do.
Working on the stage. Thank you to our panelists for joining us.